Okay, so welcome to the Society for Ecological Restoration and Northwest Chapter Hub Talk series in Missoula. Tonight we have Ylva Lekberg with us, a PhD ecologist and soil scientist with MPG Ranch, and also an adjunct professor with the University of Montana. And she is here tonight to talk to us about invader impacts on soil ecosystems. So thank you very much, Ylva, and take it away.
Well, thank you for the invitation, and thank you all for coming. It's a lot of friendly faces here, and it's a lot of fun to be here. So I often start my talks with these two iconic pictures. You might have seen them before. It's actually from Mount Sentinel, taken about 40 years apart. And so you can see like a lot of this beautiful native community has been replaced more or less by knapweed. Let's see if this works, Jeff. So I don't have to go back and forth. Try enter. Yeah, the down arrow is what I tried before. Keep pressing it. Not your fault.
No, you're good.
Anyway, you always worry about.
So just hit enter.
Enter. Okay.
When you put this like big title on, like what every restoration practitioner should know, I hope that didn't pack this room because you might walk out. I'm not going to tell you everything you need to know, but I'm going to tell you everything that I think is important to know based on what we've learned during the past 10 years. But before I do that, I just want to briefly take a moment to talk about MPG Ranch. I don't know if you have been there. We're a privately funded organization that focuses on research, education, and conservation. And we're located on about 17,000 acre property in the Bitterroot Valley. And you can come and visit us and set up tours. And if you're curious about what we do, we also have a website that you can check out. And there are actually quite a few of my colleagues here, so you can also talk to us after the presentation. And you know, we are drinking beer. It's an informal setting, so I would say like if anything is unclear as I'm going through things, just wave and we'll talk about it. We have plenty of time, so I'd rather, instead of losing you along the way, just raise your hand and we'll cover anything that is unclear as we go along. Great. Thank you. So I hadn't really thought about invaders or invasion biology until I started my job at MPG Ranch 10 years ago. And you know, the reason I work on invaders is pretty well illustrated on these maps. So this is a map of MPG Ranch, and this shows the distribution of 3 of our main invaders that are problematic in this area: cheatgrass, spurge, and knapweed. And I'm going to come back to those over and over again throughout this talk. The warmer the colors, it means the more abundant these plants are here. So you can see they are really abundant on the ranch. This is a former cattle ranch that cattle grazing ended in 2007, and it has a history of heavy disturbance and herbicide use. So that probably opened up for a lot of these invaders. Cheatgrass is actually the most abundant plant on the ranch, which is kind of sad. So this is a healthy leafy spurge invasion. We're all familiar with it here in Missoula. So I think as restoration ecologists, we have to ask, how is this site different from the native prairie? How have a lot of things changed? And furthermore, does it depend on the particular invader? If I go out to a cheatgrass invasion, do I see similar microbial communities and ecosystem processes and productivity as I would see in a leafy spurge invasion? And furthermore, like if we know that they are different, or if they're not different, how does that influence how we approach restoration? Doing a great job. So what I'm going to try to convince you today is showing a lot of graphs. It's funny, actually, last year on the ranch, we had this coach come in and tell us how you give a presentation. I'm going to break all of them. It basically says, never show a lot of graphs. I'm going to just dump graphs. But I'm going to walk you through them, so hopefully you have a good understanding. But I'm going to try to convince you that they are very different in terms of productivity. Invaders tend to increase productivity, and we about a little about why that might be. Do they all do the same things? For some processes, yes, but in other cases, it's species specific. And we're still trying to understand, like, under what conditions do they all do the same, and under what conditions is it species specific. And then finally, like, knowing that they're species specific and knowing what they are, how can that guide the restoration? Sorry, you didn't know that you signed up for this when you came.
No problem.
So anyway, so one thing that I continue to be amazed by with these invaders, and I think we all should be amazed by, is that they come in, they're naive to this, they're evolutionary naive to this environment. They come in and they outcompete and they outproduce our, evolved native plants. And one thing that is particularly interesting is that we think we have this idea of what drives productivity, that it should be the environmental conditions, nutrient availabilities, rainfall, maybe competitors, maybe some herbivory. And yet these plants that have never really been in this environment come in and they double productivity in some cases. So this is some data from Morgan McLeod. She's here. If you have specific questions, you can ask her later. It's part of her master's thesis that she did at where she went out and essentially just asked, like, do we see increased productivity among these invaders relative to native communities in this area? So she went out to cheatgrass, knapweed, spurge, and cinquefoil communities, and she basically just cut the above ground biomass in a set area, and she compared that to adjacent uninvaded native communities. So the black graphs here are all the invaded biomass, and the white ones are the native communities. Yeah.
Yes, so first question from the ethics person, but not the recreation person here. By productivity, are you meaning just biomass?
Biomass. So you basically go out and you have, I think you use 0.5 by 0.5 meters or something, you cut everything above ground, and then you weigh it. So it's very simple. But it basically allows you to ask, do they produce more biomass, given this pretty much the same conditions as adjacent uninvaded communities? And as you see, for 3 of the invaders, cheatgrass, knapweed, and spurge, yes, they do. And in terms of spurge, it's almost triple the productivity as right adjacent native communities. So then, of course, the question is, like, how can they do that? Well, we always have to be a little careful when we go out and just survey communities, right? Because these invaders might invade areas that are disturbed, have higher fertility than adjacent native communities. It may not be something that the invaders do. They might just take advantage of a better, more fertile site. So one of the first things we did on MPG Ranch is establish these experimental plots. And that allows us to ask how invaders directly change conditions. Are they able, given the same environmental conditions, to increase productivity compared to native plants that experience the same conditions? And I'm going to present a lot of data from these plots. I also want to give credit where credit is due. And most of that data was actually collected by Kelly LaFlame and Morgan. Again, so I'm just a spokesperson here. But basically what we have is knapweed, cheatgrass, sulfur cinquefoil, mixed native communities, grasses and forests, and then leafy spurge communities. We have 5 of each community type here, and they were planted in 2011. So I'm going to come back to these communities over and over because it allows us to assess causation, that the invaders actually are doing these things that we're seeing in the field or not. So one of the things that we have seen, it took a few years, but this, I believe, is data from 2017. Morgan can correct me if I'm wrong. But basically what we're seeing, again, is the productivity. Same thing as I showed earlier. So you basically go out to these small plots and we cut all the above ground biomass in like 0.5 by 0.5 meter area and we weigh it. So the taller these bars are, the more biomass is in those plots. And as you can see, the native biomass is the lowest, followed by sulfur cinquefoil, and then the 3 really great, invaders increase productivity in these areas too. So they're doing something different from native plants. They are, for whatever reason, able to take advantage of the fertility that is there, turn that into biomass, or they can increase fertility in some way. So what we're going to spend most of the time talking about today is soil biota. What is the potential role of soil biota in these changes that we're seeing? And I know that most people, hopefully all of the people in this room, don't consider soil as dirt. We know that there's healthy ecosystems, lots of processes going on. And you've seen this statistics before. In 1 teaspoon of soil, you can find 1 billion bacteria, several yards, of fungal filaments, thousands of protozoa, and up to 100 nematodes. So what are they doing, and are they changing when they're associated with an invader compared to native plants? Before I get to that, though, I feel like I should remind people about the important roles of these soil biota. And here I'm using the nitrogen cycle to do that. So nitrogen can enter the soil via at least 2 ways. One is through nitrogen fixation, where atmospheric nitrogen is just fixed by microbes, and that is strictly a bacterial process. So we have nitrogen fixation. It could also enter the soil via dead organic matter that is decomposed then by, again, fungi and bacteria. We have complex organic forms that are then transformed into inorganic nitrogen, again, a bacterial process. And then we might lose some nitrogen through denitrification. That's a bacterial process. And then plants are ultimately taking up that nitrogen, and that's often aided by fungi and bacteria. Just to give you an idea that bacteria and fungi are involved in probably the most important nutrient cycle in the soil. So how do we go about characterizing these communities? Well, we're not like botanists, and I'm not saying botany is easy, but you can actually go out, you can identify a plant, you can quantify the abundance, and you get a sense of the community. But here we're dealing with microbes, right? We can't see them with the naked eye. So before I go again to talk about the data, I just want to talk a little bit about how we go about characterizing microbes. So what we do, we often use sequencing. I'm sure you've heard the term. We either start by just taking soil cores. We go out to a community, we take a bunch of soil cores, or we use plant roots, depending on if we're interested in the microbial communities in the soil versus roots. We then use these common methods, like for DNA extraction. We extract all the DNA from either the roots or the soil. It's basically like baking. There's nothing that's so, highly advanced now, so we just follow a recipe, and in the end, you start with soil. In the end, you have a template with just DNA. It's almost like magic. Then, of course, that DNA is plant DNA, it's fungal DNA, it's bacterial DNA. So if we're interested in bacteria, we have to select that group specifically, and then we use specific primers that only amplifies bacteria. And we use a process called PCR, polymerase chain reaction, to amplify that DNA. So we have a lot of DNA that we can submit to sequencing. We now get millions of sequences back. Then we have to start dealing with. When I started this, we would clone, and we would characterize the community using maybe 40 sequences. And now with the technology advanced, we can get millions of sequences from a community. So that has presented its own challenges, but you have all these sequences, and then what we do is group them. So if there are similar sequences, they're grouped together, and then we ask, what is this sequence similar to? So then we go to global databases and match those sequences, and that's how we then say that, hey, we have these fungi, we have these bacteria, and their relative abundance we equate to the sequence numbers that we. A lot of this work is done not by me, it's done by Lorinda, so she's here too. So if you have any specific questions, you should ask her. But again, this is not me. This is a lot of other people. I understand it. I'm not good with computers, so I don't do it. But we work well together. So you can ask her about that. So I'm gonna present a lot of microbial data. Most of it has been processed by Lorinda, and I'll walk you through how to interpret it as we go along. So there she is. I wasn't sure if she was coming. So anyway, going back to the experimental plots, we wanted to know, like, after 5 or 6 years, have these invaders structured unique microbial communities. And this is what we call an ordination. It's taking a lot of data, all that sequence data, and putting it into two-dimensional space. The only thing you need to know how to interpret this is that the closer two points are together, the more similar the bacterial communities are. So in this case, you can see that the native plant and the knapweed communities harbor quite similar communities. They haven't diverged that much from each other. Whereas if we're looking at leafy spurge and cheatgrass, they're really far from each other. They're probably the most dissimilar bacterial communities. And if we're thinking about that it's plants that actually structure these bacterial communities, it's not that surprising that we see that, because you could argue that cheatgrass, for those of you who know cheatgrass and leafy spurge, they're very dissimilar, right? Cheatgrass is an annual shallow-rooted grass, whereas leafy spurge is the perennial deep-rooted form. You would be hard-pressed to find two plants in these grasslands that are more dissimilar to each other. So that obviously was going to impact the amount of carbon they allocate below ground, what traits they have, when they die, the litter quality that the bacterial community is exposed to. So it's not surprising that we see that. In fact, it's quite encouraging, because that means that the plants are structuring these bacterial communities, and there's a strong link.
Yeah, real quick. So the common garden started with the same soil community.
Yeah, exactly. You can see like how similar, how close they are together.
What was the time frame after planting?
So this, we have looked actually, you would think, given how different these plants are, that it should happen pretty quickly. But we looked 1 year after planting, 3 years after planting, and we didn't see differences. I believe that this is, like 5 years is the first time we saw it. I actually would have thought we would see it much sooner, because we saw differences in productivity, differences in soil respiration. That happened right away. But in terms of shifting bacterial communities, that took years. And I wouldn't have expected that. And the other thing is here as well, you can see how we laid this out. There are different blocks, right? So it would be problematic if we had all the leafy spurge plots down here and all the cheatgrass up here, because this is a slope. But we are having a block here, and we are randomizing them within the block. So when we're seeing these things, we know it's not because this is up here and this is down there. So, yeah. Okay, returning to Morgan's thesis, we know now from the experimental plots that they structure unique communities. And I said sometimes invaders structure unique things and they do their own thing, and it's kind of predictable based on the plant traits. And sometimes all invaders do the same thing. And we're still kind of trying to figure out when that is and when we can predict things based on plant traits. This is one example of where all invaders do the same thing. And again, like, you couldn't find more dissimilar plants, really. What Morgan did here was, again, sampling soil from these paired communities. And then she looked at the abundance of a particular bacterial group. These are called ammonia oxidizing bacteria. And they perform this important function by converting ammonium to nitrate. And nitrate is arguably the form of nitrogen that plants prefer. So if you have a lot of these, you're probably going to have a lot of nitrogen. And that's exactly nitrate. That's exactly what Morgan found. In at least 3 of these invaders, she saw higher ammonia oxidizing bacteria. So the invaders somehow seem to favor this group of bacteria that turn over nitrogen very quickly. And then the end result is also higher in 3 out of the 4 invaders. So we have higher nitrogen availability. Then, of course, could fuel that increased productivity that we're seeing. So it's almost like they're creating their own machinery here. They come in and then they change the microbial community in ways that further increase their fertility.
The zinc foil is different.
Zinc foil is different, and that's going to come up over and over again. That it's one that isn't as potent as the other one, and it's also, and you can tell me more than he probably, but I would probably argue that it's not as invasive as the other one, although that probably depends on where you go.
You said the increased respiration, did it just decompose everything?
Was that?
Did it just decompose everything? All your
Well,
reagent
that's
matter?
the interesting thing, right? Like napkin, for example, we often see higher, so soil respiration is just measuring the carbon efflux from the soil. So, and that can be due to respiration of roots, it can be due to respiration of microbes. So you just put this machine over and it measures how much CO2 is emitting from, you know this. So it's a difficult question for anyone. I get that. Now, you have to think about the whole carbon budget, right? So it's not just running it through because you also have higher productivity. So you have more soil organic matter going around. And then it's going to depend on how quickly that is turned over.
You're getting the shock of the lungs.
Yes, so they're growing a lot faster, but that also means that more carbon is going into the soil. So I don't think leaky is too problem here because we don't have enough rain. So it doesn't really, we haven't looked at this. We haven't looked to see how much we're capturing below the rain zone. There certainly could be times of the year where we're losing nitrogen, but I think in general, that probably is more of a problem in a wet climate. So we see that again in the field plots. So again, you don't know, is that because the invaders just invaded there or are they actively changing things? You also see it in the experimental plots. It is messy. You can see the variation is high. Here we've plotted the nitrate availability. So essentially nitrogen availability across these 5 different communities and 2 years. So there certainly is year-to-year variation. For example, cheatgrass had high nitrate availability in 2007, but it was quite low in 2000. And this is based on this one-time sampling in June when we go out and sample at peak productivity. So we do that every year to try to compare over years. But overall, you see that the native ones, they have lower nitrate availability in general than the invaders, which sulfur cinquefoil is somewhere in between, but definitely knapweed and spurge. They have higher productivity. They also have higher nitrate availability. So what is the importance of this? How do plants read this difference in nutrient availability? And to get at that, we worked with a high school student that was out on the ranch for a summer. She's part of that wildlife internship. We, together with Lila, we planted different seedlings. So we planted cheatgrass, so we planted cheatgrass, leafy spurge, knapweed, sulfur cinquefoil, all the 4 invaders, plus 2 native plants, a representative forb and grass. And the forb was blanket flower, and the grass was blue bunching grass. Just common grasses, common grass and forb here. Planted them in all the 25 plots. We made a little, area there where competition would be minimal. We watered them as much as we needed, so they weren't limited by water. We just wanted to see, like, we're measuring this nitrogen availability, but what do plants actually see? And does it differ between plants? Yeah. And yes, it definitely differs among plants. It also differs strongly among plant communities. So what we're plotting here is the biomass of blanket flower or blue bunch planted in all the different communities. And the different communities are indicated here. So for example, here we got blanket flower planted in native communities, cinquefoil, cheatgrass, knapweed, and spurge. And the same thing for blue bunch. What stands out here is that both native plants love spurge. Why? Probably because there's a lot of nitrogen there. And even though we measured nitrate, it was high. It was high with knapweed too, but for whatever reason, the plants can really detect the difference here. We can't say for sure that it's just nitrogen, but it's probably a good guess. But what is even more worrying is like while these grew big, cheatgrass grew enormous. So you can see the difference here on scale, it's almost 10 times different. So cheatgrass is 5,000 milligrams, that's 5 grams, whereas these guys never reached a gram. So what does that mean? Well, it means that while, you know, the native plants might like spurge soil as well, but if cheatgrass loves it even more, cheatgrass is going to win. Right? And that's what we think of as invasion meltdown. You remove one invader and another one comes in. And this is exactly, if you go out on the landscape in spurge invasions, you often see cheatgrass. And where you see cheatgrass in spurge invasion, think about that next time you go. It is enormous. And it's also really green. Because it's a nitrogen hog. It just loves that nitrogen. It's really good at taking that out and converting them to biomass. So what are the consequences of what I've told you so far for you guys, restoration practitioners? Well, know that it's probably a pretty safe bet to assume that nutrient availability is higher in invaded than native grasslands. So that's one thing to think about if you're going out and trying to restore those invaded grasslands, that nutrient availability will be high. And there are ways that you probably know better than me how to potentially deal with that increased nutrient availability. Because if you're just planting native plants there, if you have cheatgrass seeds from legacies, they will germinate and they'll outcompete any native plants you have. So one way to get around that is try to lower that nutrient availability somehow. There are various ways of doing that. Again, you probably know that better than me. One way I've heard is just dumping a lot of carbon on the soil. You can do that if it's smaller areas. Maybe you can put sawdust on that. That just kind of slurps up that nitrogen because that will increase the competitive ability of the native plants relative to the invaders. One thing that we don't know that we're hoping to learn more about in these experimental plots is how long these legacies persist. You know, we're measuring all these differences in productivity, microbial communities. But what happens when we plant a new community? Is it going to be over in like a couple of months? Or is it going to really linger those differences? Because that makes a difference too for restoration. If these legacies that these invaders are creating are long-lived, it's more problematic than if it only lasts for a few months. So that is something we don't know. The idea with the experimental plots is that we are going to have them, we've had them for 8 years now, and Morgan and I are talking about how do we move this forward? Like, can we take half of the plots and try to restore them? Just put native grass seeds or native seeds on there and see if it matters. Maybe challenge them with some invaders too and see under what conditions do the invaders win and under what conditions do the invaders win. What about anisole biota? I've talked about productivity, I've talked about bacteria. I've not come to my favorite group of soil biota, which are my personal fungi. That's why my colleagues are probably like, yeah, what took you so long? Yeah. Mycorrhizal fungi. Have you guys heard about mycorrhizal?
Right.
Yeah. Okay. Yeah. If you haven't, I'm so sorry. I had a lecture on mycorrhizal fungi when I was 22, and I thought it was the coolest thing I'd ever heard. And then I was like, that's what I'm going to do. And still to this day, I'm just so in awe by the symbiosis. That's what it is. It's mycorrhizae is the symbiosis, and it literally means fungus root. And I think there is no better picture that illustrates the symbiosis than this one. So basically, we have this little pine root or pine seedling. We have the roots that extend to here. Everything else is mycelium. So this illustrates two things with this symbiosis. First of all, this is an obligate symbiont, meaning it's completely dependent on the plant for all its carbon. If you're going to build this mycelium, it's going to cost the plant a lot of carbon. So it better do something in exchange for the plant, right? So we have carbon moving from plants, from the photosynthesis. And so this little seedling supported this whole network. There's no other way that this fungus can get carbon than from the plant. So what does the fungus do? Well, you can imagine also, for the plant's perspective, it can now explore all this soil because of the fungus. So the fungus takes up nutrients that might be here, it's going to the roots, it's exchanging it for carbon, and it's delivering that nutrient to the plant. So it's a symbiosis, meaning they live closely together. It's often a mutualism, meaning that it's beneficial for both parties. Not always, but often. What I'm going to talk about today is a specific group of mycorrhizal fungi that are called arbuscular mycorrhizal. And they form more on herbaceous plants. So they form of most of the plants that we have in this region, whereas the picture I showed before was a pine seed. So that is often on these more coniferous plants. It's microscopic, so you can't just pull up a root and say it's mycorrhizal. So that means we have to look under microscopes to make sure that they're colonized or not. The way we do that is so basically here, we can see a root, and then you can see these hyphae protruding from the roots, and then we have spores forming. So it's basically the same as in the previous picture, that these hyphae extend away from the root and help the plant acquire nutrients, and in return they get carbon. What we do to quantify how much fungi are there, or if they're there at all, is that we take a root piece and we stain it with this blue stain. Roots are not blue often, but we use the stain because fungi absorb more of this blue color than the roots do. So everything in this picture that is dark blue is actually fungal structures inside the root. So the way we quantify this colonization is that we stain these roots, and then we just look under the microscope with intercepts. It's like, are they colonized or not? Yes, no, yes, no, yes, yes. And then we get percent colonization based on total intercepts that we looked at, and then the proportion that was colonized. They're confused.
That's inside the root?
Yeah, this is inside the root. So you can imagine we take this small root segment here, we put it in the stain. This is under the microscope. And then these are all fungal structures. These are hyphae inside the root. This is where the fungus store carbon. It's basically these lipid bodies. Here are these arbuscules, are these like tree-shaped structures where the fungi delivers phosphorus to the plant. So taking a lot of time, but it's so cool. Anyway, we might not get to the herbicide portion. This is worth it.
they want.
This is worth it. Yeah.
So this may be obvious that there's a group here, but
No.
it seems to me this has a lot of implications for plants starting these carbon sinks. These mycorrhizal fungi are pulling all this carbon out there. It's going to
Yeah.
So there's so much more than just the biomass of the plant.
Exactly. Yes. Up to 30% of the carbon that is fixed in photosynthesis can be allocated to these fungi. They're going to respire a lot of it, right? And then it just goes right up to the atmosphere. But there's a lot of fungal biomass in the soil. Yeah. So what do these do? You know, this is the picture I took during my PhD because I was working with this legume called pigeon pea. Here are these non-mycorrhizal pigeon pea. So it did not have the fungi in the pot. Here they did. They were given exactly the same conditions, you know, in terms of fertilizer and water. And the mycorrhizal fungi in this little pot was able to deliver so many more nutrients to this plant to generate differences in growth like this. It is extreme. We don't always see it, but under some circumstances we can. They can also be important for improving soil structure. This is a picture I took actually from the Nyack floodplain outside Glacier. This is a root. Here you have, this is again, we're going back to these pine. This is actually cottonwood, and it forms this other mycorrhizae. I'm not going to confuse you too much, but there is a colonized root tip, and you can see this pus here. Those are hyphae. outside from this and you can, the main point of this picture is that you can see how these hyphae hold together these like sand and gravel grains. And they do the same thing in soil. So they do that physically by holding together aggregates, and they also exude this glue-like substrate that you glue these aggregates together. So improved soil aggregates stability is often a benefit from mycorrhizal fungi. Another thing that they do, we don't fully know how they do it. Min and I are looking at an analysis right now to try to figure out like how do they actually help plants tolerate drought? Because it seems like they do, but we don't know how they do it. And it's something that I think is really important for plants in this region. Anyway, this is just a picture I got from the web. There are two non-mycorrhizal plants here. Two mycorrhizal plants here. First of all, you can see that they're much bigger when they're inoculated with the mycorrhizal fungi. But the main point of this picture is that this is a drought-stressed non-mycorrhizal plant. This is a drought-stressed mycorrhizal plant. So somehow it's able to help the plant tolerate drought much more than a non-mycorrhizal plant. Yeah, we don't know how they do it, but it's pretty clear that they do. And the reason I think it's important for plants in this region is that we're starting to look at regional differences. And plants, we have a lot of phosphorus in the soil here. If you go 90, there's an exit that is called phosphate. That is death for a mycorrhizal ecologist. Because if the main benefit of these fungi is to acquire and deliver phosphorus to the plant in exchange of carbon, the plants won't need it if there's a lot of phosphorus in the soil, right? Yet, when we compare plants here, the same species, with plants in the Midwest, that where soil phosphorus availability is a lot lower, they have the same colonization. The colonization is not different, yet like plants are not phosphorus limited here, but they are there. So that's why we're starting to think maybe here, they just fulfill a different function, and maybe that function is drought. So we're starting to look at that. So how does mycorrhizal fungi relate to invasion? Well, there are two hypotheses in invasion biology that relates to mycorrhizal fungi. The first one is called the enhanced mutualism hypothesis, and that basically predicts that invasive plants are going to be more dependent and get more benefit from mycorrhizal fungi than native plants. There are some plants that fit that. Knapweed is one of them. Ray Calloway at has done some really nice work on this, showing that when you add fungicide to plants, knapweed becomes less competitive because you knock out the mycorrhizal fungi. In ecology, there's always the opposite, you know. The dogma is that it depends. So of course, we're going to have another hypothesis that states the opposite. And that is the degraded mutualism. It states that invasive plants are predominantly non-mycorrhizal, so they don't associate with these mycorrhizal fungi, meaning that when they invade, and because this is garlic mustard, it's a good example of that. All Brassicaceae fungi or plants are non-mycorrhizal. For whatever reason, they don't form this association. Garlic mustard is the brassica plant, so it doesn't even form this symbiosis. So you can imagine when it invades a lot of understory woodlands in the Midwest and the Northeast, that if you have this, it's a desert for the mycorrhizal fungi. It doesn't get any carbon from these plants. So mycorrhizal fungal abundance will decrease, and the native plants that depend on them more are going to become less competitive. So you can see that it can work in both ways. So our question was like, well, what about our plants in our region? So what we wanted to do, the first thing we did was to go out and say, how does mycorrhizal fungal abundance differ between native communities and invasive communities? And does it depend on the invasive plant? So here we targeted native communities, cheaper as leaf dispersion knapweed. And we had the same approach as Morgan did. We went out to, next slide. We went out to 6 areas in and around Missoula Valley, and in each area, we found all these communities. We found native plants, cheatgrass, leaf spurgeon. One thing that I always find so amazing with invaders is that you can find a cheatgrass invasion. 50 meters further away is an apple invasion. It's very spotty, right? And I've never figured that out. But it was good for us because that meant we can eliminate a lot of other factors that might drive what we're seeing because they're so close together. We try to sample from the same snow aspect, everything to minimize other differences. So in each of these 6 sites, we collected roots from these plants, and then we looked at, again, root colonization to look at do they associate to different extents with mycorrhizal fungi, and do they change the mycorrhizal fungal? And what we found, the first thing was, we think we have this idea of fungal richness and diversity should somewhat correlate with plant richness and diversity. If there is this sorting between some fungal species prefer some plant species. So I had thought that, yeah, maybe abundance will differ because that's all about how much carbon they're allocating below ground. But surely we should find the highest richness in native communities because we always sample from more than one plant species. Whereas going into heavy invasions, we always just targeted the specific invaders. So all invasions, we had one plant species, native plant communities, multiple plant species. But that's not what we found. You know, we looked here, you can see native mixed communities, 13 species on average per community, 14 in cheatgrass. And then you go up to knapweed and spurge, over 20. So they somehow harbored a richer AMF community, even though plant community richness was lower. And that also extended to diversity. So we had higher richness and diversity in knapweed and spurge communities, whereas cheatgrass didn't really change it, or if anything, you know, like we'll see later, it decreased it. What happened to the community composition of these mycorrhizal fungi? This is another one of these ordinations. Again, remember, the closer 2 points are together, the more similar the mycorrhizal fungal community are. So here, remember, we had 6 communities all around the Missoula and Bitterroot Valley. But here, they clustered remarkably well to the plant community we sampled from. So these are all the 6 communities, mycorrhizal fungal communities sampled from spurge communities, compared to the mycorrhizal fungi community sampled with cheatgrass invasion. And remember, these were all clustered with different sites. So you could have sites here that might be like 50 miles apart, whereas these 2 sites might be only 50 yards apart. So there's clearly a strong plant community effect here on the AMF community, suggesting that there's some sorting going on between plant communities and fungal communities. The other thing that this graph is telling us is, so we're looking at these vectors at soil and nutrient availabilities that we measured, and other things, organic matter. But again, like here you see the signal, this is nitrogen availability, is pointing right at spurge. The longer these vectors are, the more important they are, and the direction also shows where it's correlating possibly. So here it's telling me like this is really long, right? And it's going right at spurge. And that goes what we're seeing earlier in the experimental plot. Ants grow really big in spurge communities. And if you go out in the field, nitrogen availability is going to be a lot higher in spurge communities.
Am I interpreting that right,
Yeah.
though, that in the natives, the AMF communities are actually not as similar as they are?
Yeah, that's a good point. Like you can see, like there's a big difference here. Yeah, right? They seem more similar. Whereas, and I could see that you can imagine like an invader coming in. It's a monoculture. The mycorrhizal fungi are not going to be exposed to grasses and forbs. It's just going to be exposed to spurge.
But yet they're more diverse.
They're more diverse,
richer.
but they're probably more similar. You know, so that's a good point. Thanks for pointing that out.
Just following
Yeah.
up on that,
Yeah.
there's two axes, and they're actually more similar on the x-axis and less similar on the y-axis.
The native communities are
Native
talking about, yeah.
communities.
And I mean, we can look at some of these vectors and see what drives those two axes. But something we're seeing that comes up over and over again in terms of the microbial communities is that you see knapweed and natives, they're not that different, but the spurge and cheatgrass are. It's the same as we saw with the bacterial community.
Now, are you planning to throw Ventonata in your plug at some point here?
Well, I wasn't actually aware of Ventonata until like 2 years ago.
It's taking over all my personal cheatgrass communities in my house in the Bitterroot.
You know, if I were to bet anything, it's going to be over here.
Over there, yeah.
Why would AMF affect cation exchange capacity? Sorry, I know it's crazy, but like, but really.
I don't know. Maybe you can help. No?
No.
Yeah. So the cation, can you tell people what cation exchange capacity is?
Was small.
But
But I'm wondering, like, it's mineral based, so it's like the ability to hold ions or cations, sorry. But that's like a very mineral based structural.
It's mineral-based soils. We don't have.
But
organic
AMF
soils.
is doing what to the minerals?
No, this is just showing that the cation exchange capacity tends to be higher in native communities. That's all it's showing. We measure these things. That's what I was saying with nitrogen, right? Nitrogen is higher in spurge communities. Phosphorus, in this study at least, seems to be higher in cheatgrass. So, yeah. Yes.
One more question. I'm wondering if the native plant communities had a lot of asters in them, and it could be a family.
Yeah, that's a good point. Now, a lot of the native communities, as you know, are pretty degraded here. And so trying to find good, healthy native communities right adjacent to a lot of invasions, you're going to be, reduced to grass-dominated communities. So, yeah. And I think that's a legacy from all the Tordon being sprayed. It's a forb-specific herbicide. It just knocks out the native forests. So that's why you go out to a lot of native communities and it's grass dominated. You don't find a lot of forbs, and that sucks very much because forbs in general are better hosts than grasses. And I'll come back to that if I make it to the herbicide section. So just bringing that point home, like that was a field survey. We don't know if the AMF communities were different before we went out, right? And we just, for whatever reason, maybe we disperse, just invade areas that already have a different AMF community. But we don't think that is the case because we see kind of similar sorting in the experimental plots. So here it's similar ordination again, made by Lorinda. And you can see here, this is AMF communities. So you can see AMF communities associated with leafy spurge are at least on this y-axis, very different from the cheatgrass AMF communities. And again, like the knapweed and native communities are somewhat in the middle. So patterns are starting to emerge. We don't know what drives those yet. Like, is it just carbon exudation from roots? Is it plant traits? That's something we're continuing to look into. So I would argue, I started by talking about these two different hypotheses, the enhanced mutualism hypothesis and the degraded mutualism hypothesis in innovation biology. And I would argue that we have examples of both in this region. When you have, this is just looking at root colonization, but it's the same thing for abundance of AMF, so you can see this is the proportion of roots, 40% of roots are colonized by AMF among native plants. When you look at cheatgrass, it drops a lot. It's just not a good host. The main goal of cheatgrass is to grow as fast as we can for, these few months in the spring. That seed, it doesn't allocate a lot of carbon to AMF, right? It doesn't make sense. Whereas these are perennial forbs. Forbs allocate a lot of carbon to AMF, and AMF really loves these plants. They used to get higher abundance of these AMF in knapweed invasions and spurge invasions. So I think with the AMF portion, fungal abundance risks change with invasions and depend on the invader. So again, what are the consequences for restoration ecologists knowing this? So to get a little bit of this legacy question, we planted blanket flower, this beautiful native form, going into all of these communities again, and just simply asking, we know that the fungal abundance have changed, we know that the community composition has changed. Is that reflected in the same plant species that are planted in all of these communities? And in terms of abundance, it is. This was taken like 3 months after we planted blanket flower, and you can see, you know, it's a lot of noise in this, but the patterns are at least the same. So, you know, cheatgrass, blanket flower planted in cheatgrass communities are poorly colonized after 3 months, whereas in native and especially knapweed and spurge, they are highly colonized. What are the consequences of this for after these 2-3 months? You know, we don't really know. But we know that the differences that we're recording in these communities translate to a native forest that we're planting after. Again, like how long does this legacy last? Well, it lasted a couple of months at least, we know that. But does it translate to differences in community assembly over years? We don't know that. But hopefully next time we'll get this, maybe 3 years, I can tell you something.
So you plan to look at that though, to see if the
Yeah.
blanket flower increases over time or
These ones were
originally
harvested.
colonizes and then.
So these ones were harvested, so we're not following these. But what we are planning is to sacrifice half of these plots, you know, half of the size of each plot, just get rid of the invaders somehow, or the native, sprinkle seeds, and then see what happens. You know, maybe throw in some invaders there to challenge them and see if these differences that we're seeing in both nutrient availabilities, fungal communities, bacterial communities, does it even matter? Because you can imagine once you plant something that is a good host or a bad host, that's going to start structuring its own community. And so how fast do these things happen? And we really don't know. Yeah, almost done, promise. So the big question is for restoration ecologists, like, do we need to inoculate? I feel like one big message in restoration ecology that we're trying to bust is that this need of always inoculating. There are many bad things about inoculating, you know, and I feel like there's this myth that invasive plants are non-mycorrhizal or they're poor host. Therefore, you know, part of the restoration should be to reintroduce these fungi. Well, I've just showed you that if you're dealing with knapweed and spurge invasions, they have more abundant fungi, richer and more diverse AMF. Why would you ever not? So that's why I'm saying there's a lot of these products on the market, like they're calling them biofertilizers. Well, you know, maybe, but what are you actually inoculating with? There's going to be these rural AMF that come in and probably outcompete the native adapted AMF. And who knows if they can even resist. So you're most likely to just throw away a lot of money. But they're there. You should be aware of it. You should be very skeptical because I've looked at quite a few of these products, and a lot of them do not even have viable spores. So, yeah. So it should be the absolute last resort for many reasons.
So what AMF are in those?
There are these ruderal, I can tell you exactly what species mix is going to be in them. It's going to be Rhizophagus irregularis, familiformis mossy, always the same thing. They're globally distributed, they're ruderal species, they're really good at inoculating plants. Who knows if they're beneficial?
And they're both in the invasive, the abundant and in the natives? Are they
Yeah,
in both?
I don't, and that's a good question if I've even found them here, because often we
Oh.
just deal with our sequences, right?
Yeah.
So, you know, are those species here? They may be. I just actually looked at global distributions of one of those species, and it didn't cover this area. So you might actually be introducing something that isn't here, you know. And I'm going to a symposium in actually Brazil of all places. It's ridiculous. We shouldn't even travel in these days, but I okay, this is a totally different thing. But in Sweden, my native country, just came up with new words for last year and light shame was one of them.
I mean, that's fair.
It's fair. But actually, I'm going to this symposium, and one thing we're going to talk about is this issue of, creating what are the consequences? Anyway, moving forward.
That's fascinating.
So they may be, these AMS might outcompete. Could they themselves be invasive?
That's a big question. We don't know. Because we haven't been able to track them very well. So they could be, we don't know. So one way for you guys to think about if you are restoring these areas and you're thinking about like, do I need to inoculate or not? One way is just bring some roots to me. That's what it is. This is actually pretty good roots.
Oh, it's pretty.
So this is, I know, it looks kind of alien. This is from the Milltown floodplain. Like that's where
At
you
least you picked one that was colonized.
yeah, so there's still some questions,
Yeah,
they didn't know if it was
I'm
a
gonna bring you
host thing or if it was a location. But it's quite easy, I mean, you do need the chemicals and you need the microscope, but you can figure out if you have fungi there. That is the absolute first step. Are they there? If they're there, great, you don't have to do anything. If you have low abundance, there are options. You can, maybe plant something that stimulates the AMF before you do the restoration. Yeah, so you can do that if you come out to a bare place, don't have roots. It requires roots. If you don't have roots, there are ways that you can actually take soil, you bring it to me, or you can grow it. You grow it in a greenhouse, you mix the soil with sand, 1-to-1. You grow it with a good host. We often use sorghum. It's a C4 grass that allocates a lot of carbon to the fungi. You grow it at a set amount of time. Then you bring the rootstocks, and then we look and we see, like, do you actually have viable fungi in the soil? Because if you do, they should be able to colonize these plants, and then we look at colonization. So that's another way to do it. Again, inoculation should be the last because it's expensive, it's likely won't work, and you're introducing fungi you probably don't want.
Well, so how important do you think the species are that you're seeding or planting at a restoration site are to promoting colonization
so-
of whatever mycorrhizae might be there?
So there are very coarse, groupings. You have C3 grasses in general are core hosts for AMF. Others tend to be better, except if you're a grass and some other families, because they don't even form this association. So it's quite easy to find out, like, is this a good host or not, that you can plant. And thinking about also, like, is it producing a lot of biomass? Because if it's producing a lot of biomass and it gets colonized, that means it's going to allocate a lot of carbon to it. You have to think about the fungi as an obligate it's an obligate symbiont. It requires plant to get carbon. So the more carbon that is allocated below ground, the better. So if the plant grows for a long period of time, which Fortunately does, they don't go dormant. They just keep allocating carbon below ground. That's a good thing. Not that you want Fortunately, but from a microbiome. So thinking about inoculations, there are maybe times that you want to do that. And how can we do that when there's an increasing attention of actually adding native inoculants? Now, it's not very practical to go out to a pristine native grassland, disturb the heck out of it, and bring the soil to a place where you want to restore. First of all, it's too bulky, it's expensive, you can't really do it. So colleagues of mine, Dan Mummy and his group, Chuck is here. So if you're curious about this whole technique, you should talk to him and Sasha. She's not here, but what they're working on are these like turd-looking things. And they can put anything in there. Like what we have worked with Dan on is maybe putting mycorrhizal fungi in there, because that might be one way where you can actually inoculate. If you have a native grassland, you want particular fungi there, they're locally adapted, you want to put it in a place where you know you don't have any fungi. This might be one way that you do it. You basically just put it in the soil, we put seeds in there with the mycorrhizal fungi. You don't, in this case, they're trying, you can talk about it more, Chuck, but I think in this case, they're trying to work with herbicide, as well, making this seed resistant to herbicides.
Trying to find herbicide,
Yeah.
local site.
So here they're on the surface. Of course, if you're working with mycorrhizal fungi, you probably want to put them in the soil. And we just did that a month ago with flaxseed. So mycorrhizal fungi from our local inoculum and flaxseed, we put them into the soil. And we just looked at some of those roots, and they are colonized. So that if you really are at the last resort, you want to inoculate, you want to use native soil, this might be one way to make that soil go further. Because you don't need a lot. You just need the seeds that are germinating to associate with mycorrhizal fungi, and then they start spreading. So there's a lot of potential in that. Last thing I'm going to talk about, and then I'm done. Herbicides. We put a lot of herbicides on the ground, and sometimes for good reasons, sometimes for not good reasons. And it's obvious what herbicides do above ground. It kills plants. If you're using it for a specific herbicide, it kills forage, not grasses. But what I became curious about, given that plants differ so much in post quality, what are the cascading effects below ground when you're spraying whole areas that we often tend to do? So the last thing I want to talk about today is an experiment we did a few years ago where we took 3 areas on the ranch. In each area, we selected either knapweed invasion. Again, remember, they're very spotty, so they're very close to each other. So we targeted knapweed invasions, and we targeted grass-dominated native communities. And then in each site, in the knapweed invasions, we had some communities that we just sprayed with water, we didn't do anything. And then we had 3 spots or plots that we sprayed with Tordon, which is a forb-specific herbicide. We did the same thing in the grass-dominated community. Why did we spray the grass-dominated community? It's a forb-specific herbicide. We did that because we were interested in the direct effects of the herbicide. So if the herbicide directly affected the below-ground community, we could see that in the grass-dominated community. It wasn't plant-mediated. Tordon doesn't do anything to plants, right? So we just sprayed it on grasses. We didn't do anything. But if it was a direct effect, we should see it in those communities. In the knapweed communities, of course, Tordon will kill knapweed. So there we have both the direct and indirect effects. Yeah. These, I think, will be a little hard to see. We published this, so this is basically surveying the plant abundance a year after we sprayed. We went out and sprayed in August. And this is looking at abundance of knapweed in either in 2012 or 2014. So in 2012, these are the native communities. Knapweed abundance in the native communities, really low. That's good, right? We picked them because they should be grass-dominated native communities. So low knapweed abundance. Here is knapweed communities sprayed with water, knapweed communities sprayed with Tordon. We really nuked those knapweeds with Tordon. So that was a good thing. You can see in 2014 it's starting to creep up a little bit. So it has a 2-year residual. After 2 years, when I got back there 3 or 4 years after, I couldn't even see. The only reason I could see that we had sprayed was that we still had stakes. So that really makes me question, like, we go out and spray, but if you don't do anything else, 3 years later, it's going to look exactly like it did, before you sprayed. So different story. This is bulbous bluegrass. A lot of you are probably familiar with this plant. It's another exotic. It's a short-lived perennial grass, very similar to cheatgrass, but cheatgrass is an annual. When we removed knapweed with Tordon, it was replaced with Ubuntu. So this is, what we can think of as invasive meltdown. You remove one invader, another one comes. It wasn't replaced by native plants. It was just another exotic.
Harder to get rid of.
Maybe, yeah. I don't have much experience, but what we wanted to know is like, okay, we see those dramatic changes in plant communities, what are the consequences of the AMF community? So that's the last one. Before I get there though, we wanted to kind of assess host quality of these plants. We know, so we basically just went out and collected knapweed roots on the landscape. We collected some native grasses and then bluegrass. Bulbous bluegrass. And then we just looked at root colonization. How colonized are they by these fungi? And that is a good indication of how good of a host are they. And not surprising, 8 out of every 10 root pieces we looked at for knapweed under the microscope was colonized. That was only every 5th one. So 20% root colonization in native grasses. They're not particularly good hosts. But bulbous bluegrass is even worse. It's really bad host. Poorly colonized, small root system. So the fungi is not going to get a lot of carbon from those plants. So when you spray Tordon, you remove a good host and you replace it with a bad host. What happens to it? Not too surprising, nothing. So this is AMF abundance, basically mycorrhizal fungal abundance in the soil. You measure it through lipids, doesn't matter. But the bigger the graphs, the more AMF you have. So in the native communities, as we predicted, with spray Tordon, they don't respond to the herbicide because they're grasses, they're not for no shift. But in the knapweed communities, we have a drop. So the AMF abundance is less than half after 1 year. So this was 1 year after we sprayed. You have this replacement of knapweed with bulbous bluegrass. Good host is replaced by a bad host, and you have a reduction, a severe reduction. Again, if you plant a good host, it probably will recover, but it's something to keep in mind, right? When we're spraying, it's so obvious what we're doing above ground, but keep that in mind, like what happens below ground? And what are the consequences of this for restoration? And are there other alternatives? Again, like there are times where you have to spray. I'm not like necessarily anti-sprayer. I can see that there are times when you want to go out and spot spray, especially in early invasions. It's an invader that you definitely don't want on the landscape. But just think about it. Think about what the consequences are below ground and how, might this compromise restoration. And then I think I just have summaries. So I think and I hope I have convinced you that microbial communities, we look at bacterial communities, AMF communities, they differ among invaders. So they are, in some cases, invader specific and predictable. Remember, we went out to 6 different locations in and around Missoula Valley. We could pretty much predict what the AMF community was going to be with scourge invasions. They clustered very close together. And we know from the work we've done in the experimental plots that they are caused by the invader. We couldn't say that if we just went out and sampled, in the field, but because we have those experimental plots, because we're seeing similar patterns there, we can say that it's actually caused by the invader. I've also hopefully shown you and convinced you that those differences that we're seeing also associate with many ecosystem processes. We see differences in productivity, we see differences in income. Nitrogen availabilities are often higher in invaded communities. And, they can have strong legacy effects, but I'm not sure yet how long they last. That is a big question mark, I think. And so we know that these invaders have strong impacts on soil ecosystems, but if you plant a new plant there, how long are they going to last? So that's something that we're trying to find. Thank you.
Oh, I'm 100% put you on the spot here and ask you, how do you see going from the photo on my right to that? I mean, I know it's not an easy question, but I mean, just what are your initial thoughts and what you guys have learned in MPP? Where would you start?
You know what I would do? I would say we have to be patient. I think there are areas that we have to preserve. And if that means spraying, if that means hand pulling because there is a I just had a conversation at your house, you know, like there was a rare plant of the, yeah, right. So, you know, you have experience with that, that if there's a rare plant there, we probably want to protect it from knapweed, even if that means going knapweed. But when it comes like this, you know, you can't go in and spray it, you know, and you know, we know that restoration often fails. How are you going to do this? Broadcast? No, it's not going to work. Eventually, if you're taking an evolutionary approach, enemies will find this plant. Herbivores will start eating this plant. There might be another invader coming in that is problematic, but we know that from invaders that have been here for a long time. We try to speed it up by bringing in biocontrols from Europe that eat these plants, so we're bringing in enemies. That's bold. Sometimes it works. Sometimes they're catastrophic responses. Right? I mean, the cane toad example from Australia is a good example. I think, you know, whenever humans meddle in this, we can't possibly predict all the consequences when we're meddling with it. I mean, I think the work by Dee Pearson, excellent ecologist here in Missoula, have shown all these trophic cascades that we have, you know, like what happens when you bring in, you know, the biocontrol and knapweed, and then deer mice come in and eat it, and it actually starts eating native seeds as well. Like how we have a lot of controls, and before we introduce biocontrol, but we can't possibly predict all those interactions. So I think we just have to step back and say, we're going to let nature do its thing, and it might take hundreds of years, but also highlight some areas that we really care about, and those we, for whatever reason, we put our resources on. But just like the ranch has been helicopter sprayed 2 times without any like additional restoration.
With Tordon, right?
With Tordon, just sprayed. But you know, what happens? Like 3 years later, it's like you never sprayed, and you kill a lot of native forests. And you know, I think that's why actually our AMF abundance in native communities is so low, relatively speaking, because it's deep grass dominating all this Tordon levels. I mean, I don't have proof for that, but it is low, and you have these forests coming in, and it increases. I would just wait. Yeah. I think we could just screw it up more. So. I don't know.
That's nice for us.
To start, you mentioned the productivity difference between bases. Does that have any implications for the actual carbon storage?
Well, again, like you have to do a carbon budget to understand that, right? It's really complicated because, yes, they increase productivity, but often the litter is more labile, meaning that the CN ratio is lower, meaning also that they're going to turn over faster. So I think only time will tell. There are probably fancy models that we can use to predict that, but and that's something we're interested in on the ranch, you know, because obviously there are huge areas that are invaded, like, and carbon is a big deal these days. So what effect do they have on carbon emissions? And I don't know, because you can see, like, on the one hand, they increase productivity, but they also increase respiration. So what ultimately is the effect of solid organic matter? And it's such a coarse measure, and we've measured it now in the experimental gardens for 9 years, and we're not seeing differences. But I think part of that is because we're not looking at fractions of soil organic matter. When you measure that, it's so coarse. So you're not. Yeah.
General question. So it seems almost contradictory, the whole idea of the symbiotic mycorrhizae. You think they have co-evolved, yet these species from another continent can host this really rich diversity.
That's a very good
So
question.
how does that happen?
It's a very good question. The answer is that these are generalist fungi, so they're not very host specific. So these are muscular mycorrhizal fungi. With ectomycorrhizal fungi, they are more host specific. So there are certain fungal taxa associated with certain coniferous trees. And that's why, you know, until recently, we did not have any pine invasions into the southern hemisphere because we didn't have the compatible mycorrhizal fungi. Then somebody introduced this in plantations, and now they're spreading in New Zealand because they have. But for mycorrhizal fungi, you can get like 80% of plant species. I can get the same fungal species to colonize that plant. That doesn't mean that there isn't sorting going on in the plant communities in nature based on like I have these traits the fungi says well, I have these traits the plant says, and we work well together. So there's some sorting going on, but there's not host specificity. It might be some host preference. That's why these fungi are just happy that a good host comes in. They're like, I almost said that, but I'm not going to say it. They will take any carbon they can get.
Yes.
Oh, good. Yeah, that's a really good question. Thank you for bringing that up.
Just going back to, you showed pictures of showing like a plant that was colonized versus one that wasn't, and how much bigger it was. But then you're recommending not inoculating plants. So I don't.
Yeah, and that's another really good question that needs to be all paid attention. I would recommend it if there is no fungi there, but often fungi are there, or you can stimulate them increasing in abundance. So what I'm not advocating is bringing in one of those products, the fungus that I don't think is good for the plants here, and that is also very competitive and rural. It'll outcompete the other for local AMF. There are times when inoculation is good, but then that's where the whole like turd thing came in. Like if we can just go out and take native inoculum, just go out and take a little bit from native grasslands, or I don't know, maybe even knapweed or spurgeon. They have more fungi and they're more diverse, and then use that as inoculum. So you use a local source instead of bringing in. That's my only point. I think they are important. I don't think they're important for phosphorus acquisition here because we have so much phosphorus, and that's another thing that we're moving into. Because they're so colonized here, plants, and because it's such a cost of maintaining this symbiosis, it kind of makes sense that they do something. They must be doing something, because this is one thing I didn't mention is that this is a 450 million year old symbiosis. We think that these fungi help plants colonize land. So if you're that old, you cause that much carbon, you would think over time, if you're not doing anything, plants will find a way to get rid of it. Yet they're still colonized. So what does that mean? To me, it's telling me that they're doing something else. And we just have to figure out here what that means. So we're looking into drought. And I think that is one, I might be wrong, but pathogen protection is another thing that they apparently do. So that would be another, but I don't think we have a lot of pathogens here. Thank you.
Well, they just rang for last call,
Yeah.
so.
Yeah, that was a long presentation.
I mean, we can keep doing questions and socializing or whatever, but let's at least release Ylva from her stand.
Are you